the envelope the virus. So you have an envelope, and they will be fused. This process we call it a fused, and they could be directly released. It also could happen. The scenario is become an endocytosis. So they engage because it's fused with the host cell membrane system, the inside, and generate this indocin. And this indocin, usually the pH is low, 4 to 5, and then they release inside. This is very similar to the AB toxin. We talked about that. The last one is naked. So the naked, how they go inside of the body, they need those spikes. Okay, those spikes, they showed up in the host cells, and they engaged, fused, and finally they generate the indocin, and the inner RNA or DNA, the nucleic acid, is released first. Then they use host cell material to generate their capsids. Okay, so that's a very interesting process, but we want to go back to mention about this indocin. Synthesis of the viral components. We mentioned it's used cells, enzymes, and monomers. Same thing as bacteria, the nucleic acid is replicated, the gene are transcribed, and the mRNA is translated. Now, their structure we mentioned, you can see here the structure. They're not developed or translated all at a time. They have different steps. So usually the head developed first, and the nucleic acids. So that's why we call it the early proteins. And the proteiners, the structure proteins of the virus itself needed, usually they developed later, together with the collar, with the fiber, with the tail, with the base. So basically the inside, the nucleic acids, the protein they developed first, we call it the early proteins, and the later proteins also structure they developed later. This is viral release. We say it's a very simple. This is a budding. That's what I just draw there. It's very simple. You have viral nucleic capsids, then a viral matrix protein there, and it will cause hemagglutinin. What is the hemagglutinin will happen in the host cell? You can understand it when you have a hemor, that means it is a blooding. So it will cause blooding, something like an inflammation. And then the budding will be happens and finally will be released. So this is a procedure, but the key words here is the budding. That's very similar to the reproduction of the yeast. So then we're going to talk about the bacteriophage. So the bacteriophage has two different types. It could cause, which is a lytic cycle, which means it will lysis the cell. It also could be integrated their DNA into the host cell chromosome, we call it a lysogen. Because of that, we have two different type of the phage. We here give you example, is a temperate phage, is a lambda phage. So the lambda phage, they have a different developments. The first one is prophage. The phage DNA integrated into the host DNA, bacterial DNA. Then the temperate phage will choose between lytic growth and the lysogenic growth, which means you cause the burst, lytic of the bacterial cell, or their DNA or RNA integrated into the host cell. And many of the bacterial cells in the lab strains are lysogens. And this is a very good picture, showing you two different cycles. So you had a phage bacteria there. You phage attack into a bacteria. This is the bacterial chromosome. So the DNA is released first. And later on, they're using the host cell material to do the metabolic process and the biosynthesize lots of the phage, and then they release, and at the same time the bacterial cell is lysed. But lots of the time, if you have a temperate phage, what will happen? Instead of you generate a lot of their own phage, the phage genome will integrate it into the bacteria genome, become a prophage, and then divide it. This happens more frequently when they expose to the UV lights or some of the stress environment. And the more often happen for the phage to the bacteria is lysogenic cycle. That is why we have this very important concept that comes out called lysogenic conversion. This we mentioned about the diphtheria. So they talk about is that a long time ago, there is a beta phage. And the beta phage is carrying diphtheria toxin. So let's say the sequence is like that. And then they go ahead to find a bacteria genome. And then this guy goes here, it becomes part of that. And originally it is a phage, but later on it develops a toxin. And when the toxin carry on the bacteria, then they go into the host cell. This toxin still shows some of the characteristics of a virus. So they become an AB toxin. So we have a toxin domain, the transmembrane domain comes out, and this guy, remember, we have a picture talk about, they generate is the indocin. And this indocin is pH usually 4 to 5, acid environments, and then they can release. Then cause all kinds of the problem. So this theory called the lysogenic conversion explains lots of the foodborne pathogens. So we mentioned the Corynebacteria diphtheria is beta phage. Staphylococcus aureus has a toxic shock syndrome. We also mentioned about this is come from a virus. Clostridium botulinum, the botulinum toxin is from a phage. Shigella toxin or E. coli 0157H7 and the chlorotoxin for Vibrio is also coming from a phage. This is one of the theory talk about that. Just one of the hypotheses you can understand. It's more complicated in the real life, but that's one of the theory. Why E. coli 0157H7 will carry a Shigella toxin? Because originally the virus carry on the Shigella toxin, and going through the integrated with the genome, the process we call it lysogenic conversion. So the last couple of slides, we want to talk about how we're going to cultivation detection of virus. There is several other things we can do. We have rabbits, let's say animals. Then you spiked or infectious with the virus and let it growing there. We make an egg yolk. Make an egg yolk or chicken egg yolk so we can cultivate a virus there. But lots of the time, you see on the petri dish, see the bacteria growing there, or see the virus growing there. Then you will say, so let's say this is the agar plates, and you see lots of these things happen. Then you will have a question. You just said this is the obligated parasite. You need a host. But how come you can grow a virus on the agar? So you always remember when you show you the picture there, they didn't tell you. This is already have a layer of bacteria. Then they inoculate virus. And this virus could eat or destroy the bacteria. Then generate these things, we call it a plague. So, we can cultivation or numeration of virus on the agar plates with the assumption the agar plates already being cultivated with bacteria. And we could count this plague, similar to the bacterial calculation. Instead we call it CFU, we call it PFU. But the only difference is that CFU, remember the legal zone, is 30 to 300. PFU is 20 to 200. A relatively lower range. So that's one of the way people can do, can isolate and find the virus, which is a plague assay methods. I want to mention this, I forgot to say. Using bacteriophage is one of the methods to inhibit bacteria in the food industry. We do develop a lot of the commercial products is anti-listeria phage has already been commercialized. In the chicken products, we could use anti-salmonella phage. But the tricky thing here is, phage is a virus. So if you want to develop these things, you must have a listeria there. And we say the phage is an obligate parasite. So when it's anti-listeria, it's only for listeria. It's not going to work for E. coli and salmonella. And even we know listeria has Listeria monocytogenes, has enochia, has Williams-Weaver, all those different species. It works for Listeria monocytogenes, does not mean it works for the other species. Another thing we need to know, the scenario works for the phage is only the bacteria already growing. If the bacteria is not there, then you spray the phage, it's not going to work because the phage will die because there is no host. Another thing is that lots of the research has been showing these commercial phage can work is basically at 4 degrees Celsius. And because at that time, the limited amount of the listeria is there. And they are still growing, but growing slowly. If they grow very fast, the phage has to be catch the level. Because it's more specific and more targeted. So that's why we have those commercial products. But why these things will be developed? The reason is at the beginning, people think about using traditional antimicrobial chemicals like lactic acid, citric acid will generate resistant bacteria. Let's say acid-resistant bacteria. So they think if you generate a phage, they will not create antimicrobial resistant issue. However, because the phage genome could do these things, could do the integrity into the bacterial host cell, so later on, you can understand that every antimicrobial resistant issue happens as they relate to a gene. These phage could also carry on those genes. They could be transferred. They could go through transduction. Is that right? We talk about transformation, transduction, and conjugation. They could go through transduction, going through the cell-to-cell genetic communication. They will also carry on those antimicrobial genes. So instead of they will prevent that, they could be expanded the antimicrobial resistant issue. So that's why we have more research being done about those phage. Isolated phage is not difficult. It's a well-established method. But the genome and the lots of the other story inside related to the phage is more complicated. And there is could be a specific class or research projects talk about the phage. So just want to let you know, we do have those topics. It's very interesting. Only focus on the bacteriophage. So this is we talk about using plague assay to forming PFU, very similar to CFU. We also could do is animal study. The animal study, what we could do is decided, we call it a semi-quantitative, to decide the LD50, which is from the statistic standpoint, the dose will cause 50% of the animals being killed. But I said it is a statistically number. Does not mean you have 10 rabbits and 5 of them will die. They need to have multiple sets of experiments and put into the software, then run the different stimulation, and you get a number. So that's called LD50 or ID50, which is infectious dose, because 50% of the host cell infected and the 50% of the host cell died. So that's another measurement for decided to measure the virus. Okay, last 2 slides. Varroids, we mentioned. This is the RNA. Varroids is RNA. It's a single-stranded RNA. They do not encode the gene products. They require the host DNA dependent RNA polymerase to do the replication. It's called a plant disease. Palm trees in 1996 in the Philippines has been a huge damage happens related to the varroids. Now last one, we mentioned very early in the first class, we talk about some of the microorganisms. So we talk about the bacteria, we mentioned about the yeast, we talk about the virus. Virus has protein and nucleic acids. They maybe have a microorganism, doesn't have nucleic acids, only protein. That could be a microorganism. Then they call it prion. And this is a huge in the history, because everybody knows the BSE, bovine spongiform encephalopathy. That's happened related to the medical disease. Originally coming from the sheep, which is a scrapy sheep. It's a neurological symptoms. And then in 1997, that happens in the Great Britain. And the only thing what they can do is bury the infectious, the cows and the cattle. It happens very often in the United States after 2003. So, starting from 2003, we have a firewall treatment which is forbidden all those cattle products coming from Europe and of Great Britain. Because of the 2003, we do have the outbreaks here. So today, the United States beef products, if you want to import it in some country, like to China, to Australia, we used to do some business with Russia. It's not allowed to pass the border examination, you can say that. So for example, if I want to be going to buy a bag of the beef jerky and I'm shipping it to my friends in China, they will never receive it. They will receive a statement will saying, based on the 2003 the BSE outbreaks and the beef jerky is not allowed to be in the country. So it's caused a severe problem, and it also could be transmitted to the human being. Because in the human being, they will cause a very similar symptoms, which is we call it a CJD. And we also call it a Kuro in the humans. So what happened is the neurological symptoms is the muscle relaxing, and the muscle will never be contract. So when they cause the muscle relaxing, what happens is you're going to keep laughing, and you're never going to be stopped. So think about this, if you keep laughing about 2 years, 2 hours or 12 hours, what will happen? So that will finally cause neurological symptoms and paralysis of the body. So this is the end of this class. So finally we mentioned these other material related to the microorganism. Okay, so we finished talk about here, and we're going to go over the final exam questions. So how are you going to prepare that? We do offer you early exam. Will be first one is tomorrow afternoon from 12:30 to 3:30. So these questions I will be talking about these and some of them will show you the answer real quick in the afternoon. I would love to mention several questions here, which you should do, is the applied microbiology part. You should be very clear about the full name of these terminology. So, what is HACCP? We want to just write these for you to make sure you're clear about these, because these full name is important to let you know, and you could find a job in the future for these agencies. So regarding the food safety, we talk about HACCP. So what's the full name of HACCP? That is hazard analysis and the critical control points. Then what are the 7 principles of that? You need to go back to see the slides. Is that conduct the hazard analysis? So let's just see it. Conduct hazard analysis. Establish critical control points. Establish critical limits. You need to write the whole words. Okay, you can answer like that. I'm just saving the time. Okay, some people do like a couple of years ago. Establish monitoring procedures. Conduct corrective actions. Then what we do? Validation, verification procedure. And finally, what we do, record keeping and documentation. That's the 7 principles of the HACCP. Which is important. What is a gap? Good. Sorry. What is gap? Good agricultural practices. So what do we talk about that? Clean surface, safe soil. What else? Clean water. What else? Wash hands, is that right? Clean hands. That's the 4 components about that. What is a FSMA? Food Safety Modernization Act. So we call it a FSMA. So that was January 1st, 2011, become a law, and they give flexible for the small local farms. What is FDA? Food and Drug Administration. What is FSIS? Yeah, Food Safety and Inspection Services. Okay, what is CDC? Centers for Disease Control. And don't forget, and prevention. Okay, and prevention. Can critical limits of a hazard be arranged? The answer is wrong, because it has to be a fixed number. So we measure critical limits. If you do the cooking, the internal temperature needs to be 176 degrees Fahrenheit. There has to be a fixed number, cannot be arranged. What is PAA, periodic acidic gases? Why it is more frequent than chlorine? Because it used to be it's selling to European countries, especially like Russia, did not allow you to do. So why temperature control is important during scalding process? If it's too low temperature, what will happen? Your cultivation bacteria, the mesophiles, because Salmonella is mesophiles. So at 45 degrees Celsius, your cultivation bacteria. If the temperature is too high, what will happen? You're doing lipidization. So what happens is the fatty acids from the skin surface will be extracted. And finally will become organic matter and floated on the chilling tank and impacts the antimicrobial chemicals efficacy, especially like chlorine water. So just give you some of the examples. Those 4 or 5 questions are important. Okay, including some other acronym. For example, what is OLR? That is online reprocessing process. What is inside-outside brush wash? IOBW, that's inside-outside brush wash. So these things you should be look at the slides, okay? That is important. Let's look at some of these multiple choice questions. These I'm not going to go over it. I'll just give you the answer. You can study. Okay, you save a little bit of time. Which of the following is active immune response? So you have a skin, lysozyme, neutrophil, flushing. That's all non-specific. So active immune response is obviously is antibody. What is another name of immune globulin? That's antibody. So that's why we have IgG, IgA, IgD, IgM, all those type of things happens. Which of the following vaccine contain attenuated virus? What this means, and I said attenuated virus, which means is made from the live virus. So it's relatively speaking, it's most effective. 1 shot in your lifetime. Which one? That's MMR, mumps, measles, and rubella. The tetanus components of DPT vaccine given to young kids is what? We talk about this all the time. What is characteristics of DPT? It is made by chemical-like material. Toxic-like chemicals that we call, the terminology is toxoil. E. coli 0157H7 is a major hemorrhagic colitis in the United States. Is that correct? That is wrong. It's a hemorrhagic uremic syndrome. It's not a colitis. Hemorrhagic colitis, most possibly Clostridium difficile. Is that right? That's could be. The humoral immune response depends against. What means the humoral immune? That means the B cell. Okay, then all of the choices. Bacteria, bacterial toxin, and the viruses. This is the key. The humoral immune response differs from the cell-mediated immune response in that only in the humoral immune response is that. What is the major difference? You will not be confused about B and C. But you may have a question about A and D. So the first thing you need to know, the humoral immune and the cell-mediated immune. And the cell-mediated immune, we should say it's a T-cell-mediated immune. But they think you're smart, so they take out of the T. Both of them have memory cells. The only difference is humoral immune response have a relatively longer time of the memorization. The reason is they generate antibody. So secretion of the antibody is the major characteristics. the major differentiate between humoral immune and cell-mediated immune. The variable domain of the antibody, that was on the top, is that right? If you still remember, this is what's going to happen. What will happen? Heavy chain, light chain. This is the variable area. This constant area. Variable area is on the top for attaching the antigen. So binds targets antigen. So the B is the answer. Removing a person's normal flora, what will happen? It will lead into a disease, obviously, and we said about that the person's normal flora is basically your biggest immune system in the body, especially the new developed research showing that. 14. The serological reaction known as agglutination is. What is that? That is antibody-antigen binding, but more important, it is a cross-link. Okay. In gram-negative bacillus, a protein antigen associated with flagella, what is called? Remember O157H7. What does O stands for? Ceramic side chain lipopolysaccharides. What is H stands for? H antigen is flagella. And generated, the flagella created by a protein, we call it a flagellum. Okay, so the answer here is A. Okay. We just talk about the virus, we can go over these questions. The nucleic acids carried by virus consists of either DNA or RNA. We said never both. The viral capsid protein subunits are called what? We just talked about, called protomers. In an enveloped virus, the part of the virus, including the nucleic acid genome and the surrounding protein code, but not the envelope, is called what? Nucleic capsid. Is that right? Because it's encoding nucleic acid genome and the surrounding protein, so it's called nucleic capsid. It's a very basic question. Two major types of the symmetry found in the viruses. What are those two major? Well, we just talked. If you still remember, we just talked about that. We maybe go over a little bit quick, but that's why we have these questions to let you review. Right here, did you see it? Pentamers, hexamers. Is that right? So what is the answer here? Is that the answer? This is no radical. If there's no radio, it's not going to happen. It's not going to be happening. If enveloped viruses may enter the host cell by the coated. Oh, that's correct. Endocytosis, we mentioned a couple of examples. Animal cells, animal viruses can be cultivated. Well, host animals, embroiled eggs, and tissue cultures. So we talk about that, all of the choices. Hemagglutination, what will happen? Can you see what will happen? When you see hemo, H-E-M-A, what that means? It means bleeding. So we'll relate to the blood cell, the clumping of the red blood cell in the presence of viral suspension. And I said it's very similar to the inflammation process. Okay, varroils, what is varroils? A single-stranded RNA can infect some plants. Which of the following disease is caused by prions? All of these. Okay, then we have a question. What is the sequence of the events? This is what I said. That's the sequence of the events, right there. Attachment, entry, the synthesized genome with uncoating synthesis, assembly, and release. So that's steps. Okay. That's what we have. So, go over all these slides. Okay, just to be final remind you. Final extra points for the essay writing is you can prepare and then come into the exam room with only you mind a pencil or a pen and eraser to write. You cannot take notes or bring notes. If you bring notes, it becomes an open book. Okay? Make sure your handwriting we can be recognized. So you spending time to do it slowly. But other things I want to say, you don't have to do if you're already very good. This is for the students who's not doing very well to make up the points. If you already, for example, that's 360 is A, you already 300 or more than that, you don't have to do that. Okay, because when you do, we have to grading. So it depends on yourself, but if you decided to do, make sure your handwriting we can be recognized. That is important. Okay? So I'll see you next week, Tuesday, for the early exam. I'm not going to be here Friday, but Tuesday I'll be for the early exam. Okay?